Thank you so much for coming today. We're gathered on the lands of the Wurundjeri people of the Kulin Nations, and they have looked after this land for many, many thousands of years. We acknowledge their elders past and present, and we look forward to working with their emerging leaders. I'm Amanda Place. I work at the Florey Institute, and I'm the Manager of Corporate Communications and Media. I'm joined today by my colleague Dr Tom Keeble, who's our science communicator, and Professor Amy Brodtmann, who's one of our star researchers here and a neurologist. Now down to business. Associate Professor Amy Brodtmann is a stroke and cognitive neurologist at the Royal Melbourne Hospital, Austin Health and Eastern Health, and she's a clinician researcher here at the Florey. She's received many awards, and has recently won a fellowship with the NHMRC, and she's a CIA on the Dementia Research Team grant, which is a huge project. I'm sure she'll talk about that today. She focuses on imaging the brain network degenerations that can happen after a stroke, and she's looking at the way some people develop focal onset dementias. She's on the board of many important journals and organisations, and she's the founding member of the Australian Frontal Temporal Dementia Association. I'd like to welcome Amy to the stage. Thank you.
Amanda, and thank you, Tom, and thank you everyone for arriving here on this slightly strange Melbourne day. We should be used to it, but we're not. So what my brief was to talk to you all a little bit about healthy brain ageing, and actually how do we essentially have a normal ageing process. What's normal for the brain? What I wanted to speak a little bit about is really to update you, to talk a bit about the risk factors for bad disease or brain disease, and also what you can do to prevent it. And I'm just going to focus on a couple of things. So I think that one of the issues is that there is bad news, and the bad news is that things that we do in our life actually cause accelerated brain ageing. There's many lifestyle factors that can cause accelerated brain ageing, and they are essentially the same things as vascular risk factors. And you would have heard about some of these: smoking, diabetes, high blood pressure. When we look at our brain, there's lots of different ways that we can test to see whether we have normal brain ageing or accelerated brain ageing, and I'll speak a little bit about that because they can manifest as two lots of changes. It can manifest as changes we can see on brain scans, which is actually with atrophy, with white matter change. And atrophy is a term I'll use a bit in this talk, and it's actually what we refer to as brain shrinkage, so shrinkage of the brain. We can also detect these changes when we examine someone neurologically, and we can detect them with our cognitive tests as well, because the brain is just another end organ, and cognitive decline, if you like, is brain failure. So vascular risk factors cause stroke. Stroke and dementia are linked, so I will talk a little bit about the fact that there is an association with stroke and dementia, but it's not inexorable. There's things that we can do, and I suppose the good news is that there are ways of actually altering our trajectories. It's not always downhill. So one of the things I wanted to mention is that people often ask me, what is dementia? And I think that there's a consideration in the community that dementia is something that's normal with ageing. It's not. It's something that is always associated with memory loss. It's not. And I think that the term Alzheimer's disease, which is a form of dementia, characterised by particular pathological proteins in the brain, is the only type of dementia, and it's not. There's lots of ways that we can actually have dementia syndromes, and when we define dementia, all it means is a progressive decline in our cognitive function leading to loss of ability. So it's starting to affect our ability to walk, talk, to interact, to manage our financial affairs, to deal with our social functions. It's often separated into early and late onset, and for this talk I'm really just talking about late onset dementia, because early onset dementia is, whilst there's an overlap in the conditions, there's actually quite a difference in the pathologies that cause it. There's a lot more genetic causes for early onset, Early onset's often from traumatic brain injury, from people who've had drug and alcohol abuse, and from a whole range of pretty rare conditions. So I'm talking about late onset dementia, and that the characterisation of that is before or after the age of 65. So it's not that late. Again, I'm not going to go into the different types because I think that we could be here all day, and I have spoken about this in public lectures previously, but people often think it's just memory loss, and it's not just memory loss. That's something that's characteristic of Alzheimer's disease, but there's many other functions that can be affected. It's always associated with accelerated brain ageing, and in fact the hallmark, the characteristic feature of neurodegeneration, which brain diseases often have, is brain shrinkage or atrophy. And so it's actually a way, brain imaging, and you saw some beautiful pictures from the magnets out at the Austin, which is where I'm based at the Austin Florey campus, showing that we can actually image the brain with really advanced methods now. So the thing that you need to know is that atrophy is seen in association with all vascular risk factors as well. So I've lifted it's hard to see because it's actually quite a large figure, but I've lifted a figure from the Lancet Commission that was published last year, and I'd encourage you, this is actually a publicly available document. It's large and it's scientifically written, but it is a remarkable document where they've basically canvassed opinion from a range of people to look at how can we prevent and how can we manage and identify dementia. And one of the interesting things from that is that the risk factors for dementia were a little bit different from what I was taught at medical school. I was taught that it was, largely a family history, and that perhaps, having not much education or head injury when you were younger were your risk factors. Well, we know that in fact most of the risk factors for late onset dementia are vascular. So as I've said here, hypertension, smoking, late onset or type 2 diabetes or adult onset, obesity, which is associated with a high BMI, which is a body mass index, a way of measuring your height and your weight. And there's no doubt that we're all too fat as a society. But the things that also came out was, in fact, early life lack of education was a potentially modifiable risk factor. So not only as a society can we intervene to make sure that our people are educated, but also we can educate ourselves. So we should be maintaining that cognitive input throughout our lives. So learning a new language at 90 is a great thing to do. Learning your 5th language or your 10th language at 90 is even better. But one of the things that was very interesting was that hearing loss came out of this large study, and it's difficult to know whether or not hearing loss is associated with ageing or whether it's associated with neurodegeneration. But in any event, it's potentially modifiable because certainly by getting checked and having that identified, we'll get more input. Social isolation and lack of having this intellectual and social input is also a potentially modifiable risk factor. And I suppose that hearing loss ties in. We are social creatures, we are very social creatures. And I think that we had a recent family holiday to Tanzania where I went on safari for the very first time. And watching all those mammals as groups, extraordinary biomass, really made me remember that we have evolved to be extraordinarily social. We need people, we need to be with people, we need to interact with people. It's good for our brains. So if we can't hear them, we need to get that fixed, and we need to be with them as well. The last thing is ApoE4 status, which is a genetic variation that we all have. We all have ApoE in our blood, but we have one of 3 alleles, 2, 3 or 4. And it's always, as I said, it's important to realise there is a small genetic risk, but on this particular graph it was, and as you see it's a tiny figure, it was only 7% of the risk. So only 7% of your risk is actually predetermined. So when I put up slides and talk about the risk factors for stroke, you'll see that they're essentially the same. So they're hypertension, smoking, type 2 diabetes, raised blood lipids and cholesterol, obesity, lack of physical activity. Now, I didn't talk about that in the first slide because that's something I'm going to be speaking more about right at the end, because not only are we too fat, we're too unfit, and we should all be walking, moving, running, swimming, rowing every day, dancing. Atrial fibrillation is another risk factor that's particularly for stroke, as well as this particular ApoE4, which is important. So when I talk about the brain, and when I talk about how could it be that, why would vascular risk factors actually be affecting us, why would it be important, I think it's just important to review the blood supply to the brain. Why would it affect cognition? So this is a fantastic slide from a human brain it's actually a slice probably from about in between our ears, and where you can see the blood vessels in the brain. I'm sorry, I'm going to have to step back for some of it to point. You can see the blood vessels in the brain have actually been filled in with a little gel. They've actually been perfused after death. And these blood vessels here are the ones that come up from the neck and actually branch out into the brain. What's important to look at is that these blood vessels are quite medium-sized, as are some of the ones that are coming on the outside of our brain. But the majority of the brain, the majority of the inside of the brain, is perfused by these tiny, fine blood vessels, which are called long penetrating vessels. These vessels that are incredibly vulnerable to things like high blood pressure, especially, as well as things like type 2 diabetes. They get hardened and thickened with these diseases. And so when we think about these blood vessels, it's not surprising that they could be affected by conditions where we're getting thickening and hardening from cholesterol in the vessels coming up from the neck, causing a blockage to the brain, and a stroke, or an infarct is the term. We can get hardening and thickening of the blood vessels, or a clot that's flicking up from the heart, causing a blockage to the blood supply of these smaller blood vessels, the branches that are going out into the brain. But also these small blood vessels can both block and bleed. So we can have hardening and thickening of the tiny little vessels, which means that they're actually developing, they can block, and little parts of the brain in there can die, as well as becoming more vulnerable and more fragile and bleeding. So all of this condition, the white matter and the grey matter, is in fact absolutely dependent on the plumbing, because if the plumbing's bad, the brain can't get blood and oxygen and nutrients, in which case it can't survive. And the reason why these fine vessels are so important and this white matter is so important is, if we think again, so this is that same slice that I showed you before, these fine vessels here are in fact perfusing some deep structures within the brain, where the spinal cord and the brainstem actually come up and plug into the brain. And there's these deep structures in the middle of the brain, which are the thalami and the basal ganglia. They're long names, but they're in fact essential for all of our structures. They're the ones that in fact are the gateways to all of our cortex. So all of our cortex in fact is talking to these deeper structures. And these deeper structures are the ones that are making you stay awake while I'm here, if you are. These are the ones that are making you pay attention, that are allocating attention to me when I'm talking, to the slides when I'm not, or to someone's mobile phone if it's going off. That's what we do. So the substrate of all human brain function is in fact attention and the ability to be awake. And when I teach medical students and neurologists, I often do this little simple model of cognition, this pyramid of cognition. We think of our human brain functions as being our ability to speak and our ability to remember and our ability to judge, have insight, be creative. But they're all based on the ability, we have to be awake and we have to attend. So in fact, attention and arousal are the substrates. And if you think that those tiny fine blood vessels are the things that are actually keeping us awake and on task, you can see how important they are. So when people come to these talks, or when my patients ask me, tell me a little bit about the links between vascular disease and stroke, and do they actually cause dementia? Well, yes and no, I suppose, is the answer. We know that brain volume loss is associated, so that atrophy, that neurodegeneration, that hallmark of accelerated brain ageing, is associated with all the vascular risk factors that I mentioned before. So it's been described with midlife and untreated hypertension. There's one study now showing that treatment probably helps. That it's associated with type 2 diabetes. So again, you can hear that there's the same risk factors are coming up. It's associated with high midlife body mass index. It appears that whilst being overweight when you're younger is bad, being overweight in midlife is particularly bad. If you've been overweight all your life and then start to lose weight at the end of your life, it's actually a bad thing too. So we need to be, if anything, we need to be fat when we're 80. So eat chocolates when you get to your 9th and 10th decades. I don't know if that's entirely true, but we'll see. So things like smoking are a very, very strong risk factor for accelerated brain aging, probably one of the strongest, but so are low levels of physical activity. And I'll talk about that just in a moment. And in fact, in the Lancet Commission and also a vascular risk factor Australian review that came out last year, smoking and lack of physical activity had the same risk for dementia and almost the same risk for stroke. So incredibly strong risk. So not running around is the same risk as smoking. And I think that we often underestimate that when we can't be bothered getting up and moving. So it's no surprise then that stroke and dementia are linked. And in fact, when on the stroke wards, up to 1 in 3 people have a history of some cognitive decline at the time of the stroke. So they'll come in and the family member might say, yeah, she's well, she's living in the community, but she hasn't been herself and she hasn't actually been able to do her banking recently, or she's needed a bit more help around the house, or she's stopped driving. And so it's no surprise it might be a history, not necessarily of dementia, but of cognitive decline. What's, I suppose, more concerning is that up to 1 in 3, again another 30% of stroke patients at 5 years have a diagnosis of dementia. Now these might be the people who are already declining beforehand, but there's no doubt that stroke is having an effect. And I'll show some data from the Canvas study that I know some of you have heard about and some of you actually have participated in, showing that there is this quite strong link. But what hasn't been understood is it the stroke that's actually causing our brains to atrophy and to have disease following the stroke? Is it just a single event? Or is it a combination of those vascular risk factors leading to cognitive decline and that they're co-varying together? Or is it both? Do they both have an independent effect? One of the things that's interesting after a stroke is that we're seeing that there's a lot of decline in brain regions that aren't actually affected by the stroke. And again, these are in some deep brain regions. These are in the thalami, those gateways, those areas where the brain plugs in, but also in a brain region called the hippocampus, which is incredibly important for memory and is a very dynamic structure in the brain. So one of the questions that we've been trying to answer here at the Florey is it from the stroke or is it from vascular risk factors? But one of the things that we've been able to do in the Canvas study, in Canvas we're looking at a group of people who had no cognitive problems at the time of their stroke, and we're following them from their stroke for a period of now 5 years. And we're wanting to see which of these people develop cognitive problems, who doesn't and why, and also to see whether or not there's increased brain ageing in this group of people compared to a group of healthy controls. So we've got healthy people with no stroke, they do have some vascular risk factors, and we've got people who have stroke. We published a paper last year in a journal called Neurology, and it was Emilio Werden, who's a postdoc in my group, where we actually showed that even at the time of stroke, because presumably of vascular risk factors, the brain volume is already compromised. And this is a group, and if you have a look at the group, you'll see that there's quite a wide spread. So this is total brain volume, the whole brain actually being measured, and you can see that there's people in the stroke groups that in fact have brain volume just the same as the controls, but that as a group there's certain structures that particularly are declining. And these are these memory structures, the hippocampi, as well as the total brain volume. So when we looked at our controls, our healthy people with no stroke versus our people who presented, who first ever stroke, this is the first time they'd been in hospital with a stroke, versus a small group of people who'd had multiple strokes and were in the study, we could see almost a stepwise decline, suggesting that not only the vascular risk factors, but also the stroke were causing problems. Oh, sorry. And this is a slide that this is a figure we didn't actually include in the study, in the paper, but that I particularly like, where the controls are over here, all clustering around a median and all slightly higher, mostly slightly higher than our stroke people, but that overall our stroke patients' brain volume was reduced in this region, but that our recurrent strokes, here shown in red, were in fact below even the median. So it suggests this dose-related, the combination of the event as well as the vascular risk factors. And so we've followed these people now, and these are some graphs of people who've been followed for a year following their stroke. We've finished our 3-year follow-up and we're writing that up now. And you can see that in some selected brain regions that I've shown in these slides, stroke patients are in red, controls are in green, that there is a reduction in brain volume far greater than you'd expect for our controls. And what's interesting is that this hatched area is in fact in the first decline in the first couple of months versus decline over a year. So you can see that most of the decline, at least 50%, is actually occurring just after the stroke in the first couple of months, suggesting that there's an inflammatory cascade that's actually causing brain volume loss over that time that stabilises. So the combination of these vascular risk factors and the stroke, it's a bad thing. One of the other projects that we're running through this dementia research team grant that Amanda mentioned is actually a study where we're looking at people without stroke but with type 2 diabetes. And we're recruiting people over the age of 50 to come in and we look at their brains, we look at their arteries, and we also look at their hearts. We're particularly interested in some heart thickness measures as well as their brains. And interestingly, when we started, we just looked at some provisional analysis to see whether or not our people with diabetes had comparable brain volumes to our controls. These are people without stroke, no stroke. You can see that in certain regions, such as, they've got long names, the amygdala and the accumbens, they in fact got smaller brain volumes than our controls. So they're age matched and sex matched. The only difference is diabetes versus no diabetes. What's interesting about these structures is that these are really important for our motivation and drive. And it's concerning that people with diabetes are actually having effects in their motivation and drive centres, because one of the big problems with diabetes, type 2 diabetes, is compliance. People don't really want to take the drugs, they can't see that it matters, they don't see how high sugars actually can affect you. And the fact is that it's affecting your brain already. So people without dementia, without stroke. So everyone looks grim at this point in time. Everyone's big-eyeing at me and looking and saying, this is terrible with this woman, please be quiet. And I suppose the question, when I gave a public lecture some years ago and left it at that, the questions were all, what can I do to prevent it from happening to me? And obviously we've got our panel members who will be speaking about what they've done to prevent it from happening to them. But I suppose one of the things, and this is actually a slide that I made some years ago, is you can do a lot. You can get checked, you can get lean, and you can get fit. So when I say you can get checked for diabetes, you can get checked for hypertension, you can get checked for cholesterol, you can get your hearing checked to make sure that you're actually wearing the right hearing aid and that you can hear things, that you can be enriched by all this marvellous social interaction that's so important for us as human beings. We can get lean, and obviously that's easier said than done, but we all need to be adhering to a Mediterranean diet and probably doing our best, and certainly getting fit will help. With our leanness, although I'm not proposing a Biggest Loser program, I'm just saying that, do more. And I suppose, again, these are a table and a figure from that Lancet Commission, which I'd encourage you all to have a look at, where they showed the relative risk of each of these behaviours, and also showed a figure which was brain mechanisms, if you like, for preventative strategies. I think it's quite good. And you can see in the middle, it's exercise. Right in the middle is exercise, because it overlaps on our intellectual and social stimulation, it overlaps on reducing our inflammation in the brain, which is probably really important, and it overlaps on all of those other risk factors, which are obesity, diabetes, and cholesterol. All those can be fixed, even cured to some extent. Hypertension is certainly not easily curable, but it can be managed better if we're very, very fit. So I really encourage you all, get checked, get lean, get fit. So Because one of the things that we really found in all of the retrospective studies that have been shown in the last 20 years is that there's a strong correlation with low activity and dementia. And there's also a strong correlation with high activity and lack of dementia. Sometimes we don't always see that in scientific studies. Low levels of midlife activity, that is being inactive in our midlife, if we maintain that to our later life, is in fact a strong risk factor for the development of dementia. And this is a study that's been done in 20,000 people in Northern Europe. And the opposite is also true. Higher levels of midlife activity being maintained to later life, in fact, reduces our risk of dementia. So I think that we all should get started very soon and continue it until the very end. We actually found this interestingly in our Canvas study, in our study of people with stroke. So one of the things that we did in Canvas is we fitted them with an armband, and they had to wear that for 7 days. And it actually allowed us to monitor their daily physical activity. We weren't exercising them at this point, we were just seeing what they did. And we saw a strong correlation with their levels of daily activity, just getting up, walking, being upright, being out, and their ability to pay attention. So when we measured them with pen and paper tasks, we saw that they in fact had better levels of attention. But we also found a very interesting correlation when we measured a way of looking at brain connections and when we looked at brain connectivity. And this is a paper that we published last year with Prof Brodtmann, who was a postdoc in my group. And we found that areas of the brain were better connected, particularly in the attention networks. And this was an important study because it was one of the very first correlations of physical activity with attention or cognitive networks after stroke. And it's really biologically valid because it really correlates with the literature. There's no doubt that attention is boosted by our ability, by our exercise. I don't know if any of you have ever gone for, felt a bit tired and gone for a run. Certainly students use this all the time when they're studying. Not so much in my time. I think people used to use other forms of stimulation, but people run now. I think one of the things is that exercise is really this neglected prescription. There's lots of evidence that physical activity is good for us. It's good for our brains. So in December of 2016, we actually launched a study which is the post-ischemic stroke cardiovascular exercise study, or PICES, and we're trying to compare 2 exercise interventions after stroke, getting people 2 months after stroke when they've gone through their rehab and actually putting them in and comparing 2 lots of exercise to see which is better. And our aim, our outcome measure, is actually to see if it reduces brain shrinkage, to see whether or not we can stop their brains from shrinking abnormally after stroke. So I think to summarise, vascular risk factors are risk factors for dementia and they're risk factors for stroke. They're bad for our brains. Stroke and dementia are linked, but there's a lot of evidence that dementia is preventable. And I think that we all have to get checked, get lean, get fit. Intervening with aerobic exercise after stroke might actually prevent cognitive decline, and we're investigating that right now, a study that's been done here at the Florey. And in fact, exercise might stop your brain from shrinking. So I thought I'd give you some examples, because people always say, oh, well, I can't do anything now, it's too late. So this is Nanamal, and she's a woman who teaches yoga, 100 students a week in India, and this is a woman called Tao Porchin Lynch, who last year was a star of a Athleta campaign, which is actually gym clothing. The top is me after a yoga session in Kerala some years ago with my yoga teacher there, who was a mere 78. He didn't start teaching yoga until he stopped teaching as a high school teacher and then learnt to do yoga, and he could do things that I've never seen any other yoga teacher do. So he was remarkable, only 78, mere child. Nanamal and Tao are both 98. Both 98, and to my knowledge, these were pictures from last year, but they're both, so you can see there she's doing a shoulder stand, and she's doing plough position, and I think that most people would be happy to be able to do that. 98, no excuses. And these are 2 people that I've always put up right at the end, because these are 2 people who are doing cardiovascular exercise, and this is Ed, who did a sub 4-hour marathon at the age of 82. And which is not bad. She's not bad. And he's still running. I think that's his 14th marathon at that stage. And there's Harriet, who did a longer marathon, 7 hours, but she's 91 and she's still running, and I think she's 94 now. So I think no excuses. We just need to move. Thank you.
How inspiring after what was a pretty worrying forecast there. I'd like to invite Molly and Ian up to the table, if you wouldn't mind. Molly Haywood is in her late 80s and she's been a member of my family for many, many years. We're incredibly proud of her. Molly has recently moved from living in Mitcham on a great big block of land with a gorgeous garden that she maintained, and she has recently moved inner city, and I'll let her tell you about that in a moment. An absolute inspiration. And we also have Ian Pearson here. Ian is 84. Is that okay that I told everyone that? Ian was a Victorian Football League footballer back in the 50s. He played for St Kilda and Hawthorn. And in 2013, Ian had a stroke and he was in rehab, and he was just telling me that while he was in rehab, he had his iPad with him, and part of his rehab was to learn how to operate the iPad very effectively because he had to keep an eye on everyone at his travel agency. And he was keeping an eye on the finances and how they were doing. So I'm sure it'll be fun to hear from him as well. Molly, we might start with you if we could. Why did you decide to move to the inner city from Mitcham?
I had a large garden, nearly a third of an acre, and that kept me fairly busy. But some of the tasks were getting a bit beyond me. My daughters had put a ban on my climbing trees. And I needed to be nearer to the family as I got older, and it meant that, as it was, I was driving in on the freeway whenever I came in to see them. So I started thinking 2 or 3 years in advance about what I should do and how to set about it. And eventually sold my house a year ago, and earlier this year found another one, a smaller one, a terrace house, and limited garden, unfortunately, but I'm learning how to grow more things in pots than I've ever done before.
Ian, I wonder if we could ask you, after your stroke, it must have been terrifying, I'd imagine, after being so fit for so long and independent. Tell us about your journey back from that stroke.
Well, initially when I had the stroke, I didn't know what it was. I got up in the morning, put my watch on back the front, jumper on back the front. My wife saw there was something wrong, immediately contacted the GP, and I finished up in the Austin Hospital that evening. And after, I only stopped overnight, but they did a lot of tests and what have you. Went to rehab for 3 weeks. I virtually had one occupational therapist looking after me virtually most of the time. I thought she was very strict at the time. I thought, yes, but then now I realise what she was really working on. My stroke affected my vision on the lower left, and everything she did, I always had to look ahead and just move my eyes to the left. She'd take me walking out in the street, ask me a question about a house across the road, and of course and she immediately jumped on me, and I had to move my eyes and tell her what it was. I lost my licence for, naturally, the GP immediately contacted the people, and I thought, now I've got to really work on this, and for 3 weeks it was all exercise, particularly with ball exercise, throwing to the left, catching a netball, which wasn't particularly hard for me because I had football instincts. And a lot of the walking machine, that was a lot. Walking in a straight line, which was difficult initially. After I left the rehab after 3 weeks, I had to continue on to the Repat hospital, to an occupational therapist again, probably for the next 4 months, probably once a month. And then after that, I thought it might be time to ask her if I could go for a driving licence. Again, she arranged and took me out for a test for the licence, of which I was successful in getting it back. And
Ian, I'm told that you are now back to being cognitively and physically as fit as you were before the stroke.
Yes,
It's pretty amazing, isn't it?
It's
Long road back.
I actually, when I had the stroke, initially after it, when I went into rehab, I was definitely the fittest one in the hospital.
And Ian's also part of the Canvas study that Amy mentioned, and I think you said he's one of your star outcomes. So congratulations! A lot of hard work there, and you've come back.
I did it for 4 years, and I think during that period they asked me if they could put radiation into my blood system, and the GP said to me, you get more out of normal air anyhow. So I never got the result from that, actually.
He can give you that, that's fine. So one of the things that we did do is we looked at, and we're still analysing these data, so most of the study had no radiation, I have to say. But there was one component where we invited people to see whether they wanted to have a PET scan. I don't know whether you've heard about that, but it's a way of imaging either brain function or brain proteins. So we can see whether one of the proteins that's associated with Alzheimer's disease, amyloid, is in the brain. And we've looked at to see whether or not stroke increases your risk of having amyloid in the brain. And there was a small number of people who participated in that study, and it doesn't look like that is the case. We're analysing those results now, though.
I have a question, Amanda.
Yep, sure.
Amy put up some amazing pictures of the yogis
Yes.
in various contorted poses.
Yes.
I imagine that might be a bit beyond many of us in the room, if not most of us. Molly and Ian, perhaps Molly first, what have you done during your lives to keep yourselves moving? How have you kept active?
When I was younger, I used to play tennis and go bushwalking. When I was about 40, I suppose, I decided that I'd join my husband at a gym, and I've been going to a gym pretty regularly, apart from trips overseas and things, for 45 years, I suppose, on a pretty regular basis. I haven't been so good this last year because I was involved in packing up the house and doing things like that, and I hurt my knee at one stage, and that ruled me out of some of the cardio things.
And Ian, I understand you've been very active your entire life. What's your history?
Yeah, so once I finished football, I played a lot of squash and social tennis. Over the years, my granddaughters, I spent a lot of time going and watching them play netball. One of them's in a squad for possible Vixens eventually, which is a Victorian professional netball team. I still work. My wife and I own a travel agency. I won't say where because I'll be advertising, but I go to work 4.5 days a week. I don't consult anymore. I just do all the accounting errors that the consultants make. It's all back office accounting, but I've always been an accountant. I wasn't a qualified accountant, but I've done accounting. I used to work for the taxation department, and I've always done accounting from leaving school, actually. I'm still working. They get me up out of the desk because I sit down a lot at lunchtime, send me out walking, and that's an effort, but it's worth it because it's around Eltham. It's very hilly. I've just told you where it is. But also I'm lucky that my street runs down to the Yarra River, and there's a marvellous walking track around the river, and I utilise that as much as I can. In fact, from the day one when I had the stroke, exercise. Every time I go to my GP, how much walking have you done? So it's kept in there all the time that I must be doing it.
I'll ask one more question of both of you. I wonder if I could ask you both to speak about your community involvement and how you are staying in touch with society and, obviously through work that's happening, Ian, but Molly, what are you doing? And Ian, Ben, I'd like to hear from you too.
Well, when I retired, I decided I didn't want to vegetate, and my husband and I travelled for about 6 months and were pretty active during that time. Then I came home, and I'd always been involved in some church activities, and I continued with that. And I was a friend of the zoo, and for 25 years I worked as a volunteer at Melbourne Zoo, which was really interesting. Anything from greeting people, giving instructions as to where to find animals, and taking part in observing animals and reporting on that. So that was a big part of my life. When I decided to give that away, I joined, or before that actually, I'd been involved in other groups. The Friends of the Library, Whitehorse Manningham Council have a Friends of the Library group which is involved in running book sales to organise funds which are then paid back to the various libraries for programs for children in particular and for adults. I was involved with a drop-in centre where people came, often depressed people, just wanting companionship, someone to talk to, have a cup of tea or coffee, get some food. We had a food bank. I belonged to the gym. I used to go regularly 3 times a week. And I was also involved in an op shop where I was at the church. And since my move, I've become involved with another very multinational church in Brunswick, where there are 18 different nationalities, and I'm involved in the op shop there. And there's an interesting mix of people coming in. People from on the street who haven't got any money, drug addicts asking for money, which we don't give out. We'll give food vouchers and help them in other ways. I was part of a community garden for 23 years. We grow all our own vegetables in a community garden, and digging got a bit heavy, so I restricted my activities to growing what I could at home. And besides, I was producing too much in the way of food, and my grandsons couldn't cope with all I was producing.
Thanks, Molly, that's great. And Ian, do you want to talk about community involvement, I suppose, the football side of things?
Yes, well, when I finished playing football, I was appointed manager of the Hawthorn Reserves, and that involved me for about 6 years until I got married, which sort of cut it on. I had to devote more time to my family, and both Hawthorn and St Kilda Football Club have got very strong past players associations. I go to a Hawthorn luncheon every month that they hold, and they have excellent speakers. St Kilda have the similar. Always the first match of the year, Lindsay Fox opens his home in Toorak. He is an ex-player at St Kilda, and he invites us to lunch at a barbecue lunch, and he's always got coaches to take us to the first match of the year. After that, I have been on the committee of the Chamber of Commerce in Eltham for quite a few years. I've I try to stop active. I've done a lot of travelling. In the last 3 years, I've done 3 cruises in the Mediterranean, Baltic and around there with my wife. And we're very, naturally, because we have a travel agency, we're very interested in travelling as much as we can before we retire. So